molecular systems that we are interested in describing sort of larger scale kind of phenomena at the level of coarse-grained representation. But then very often you're interested in knowing atomistic details in those coarse-grained or in those molecular systems. We will not be able to describe large structural changes at an atomistic level. That's why we go to a coarse-grained model. But once you have seen something that might be relevant, you have seen the aggregation, you have described how these proteins come together, something that you cannot do atomistically, you want to be able to go back and do atomistic representation of those molecular system and describe and understand what might be happening in those systems. So that's actually exactly the case. So let me actually skip this one. This is not really relevant, just to give you an idea. So even at the level of coarse-grained description, we have different representation for the side groups, for the side chains of this protein. And we can see that interaction between certain type of polar side chains, these green guys, is the main drive for the structure of the aggregate that we see in here. So we can describe certain phenomena, for example, insertion of the helix into the membrane, which is very difficult to describe atomistically because it's a slow process. Is nicely captured in these simulations. Again, time scale should be taken with a large grain of salt here in this case, because if you do 120 nanosecond atomistic simulation, you wouldn't see this transition. So these time scales are not really the real time scale associated with the transition in real world, but we can qualitatively describe them. You can see the helix actually goes across the membrane, comes to the surface, and actually in some cases goes back, which nicely agrees with experimental data suggesting that you have a mixture of different states for this protein in the membrane. So we can see actually how it comes to the surface of the membrane. We even have the separation of this from the membrane, insertion into the head group region, and complete insertion into the bilayer, completely described at the level of coarse graining. We have transition between these two. You can start looking at the kinetic properties again to the degree that you can trust these time scales, but at least you can get some qualitative behavior. Some of the data that we got, of course, it's very important to compare the experiment and see how much these models mean and are reasonable. So based on these simulations, actually for the first time, you know, the classical model in the literature was really nicely ordered helical bundles of these antimicrobial peptides. Whereas the model we observed, if you go back and look at here, you have a very disordered kind of system. In some cases, they form aggregates, not very regular, but in most cases, they have all sorts of states in the bilayer. And that seems to be more consistent with what is known experimentally. If you look at the tilt angle of this guy, for example, this helix, and look at the angle of the axis of the helix with respect to the membrane normal, experimentally, you have such a distribution based on NMR. And if you look at the result that we got from our MD simulation to reproduce that kind of experimental property, you can see a disordered kind of configuration of this molecule explains or captures the experimentally observed property very, very, very closely. So after this work, we actually suggested that what people think is not really true. Most of the time, we have a sort of tilted and highly disordered structure of the peptide in the membrane. But again, the question is, how does such a configuration in the membrane can kill the cell? So in this model, people suggest that you form a nice regular channel. Now, the membrane potential, which is composed of proton gradient and ion gradient across the membrane, will be killed because these ions can dissipate through the pore formed by these guys. How can we explain the antimicrobial effect of this peptide based on the sort of disordered configuration that we characterize using our coarse-grained simulations? So that was the next step, and that was something that we could not really do at the level of coarse-grained. These are some snapshots taken from the coarse-grained system in terms of how the aggregates come together and how they interact, but we didn't really see anything going across these membranes and these peptides. So what is it? How can we perturb the permeability of the membrane by this irregular configuration? So that's exactly the point. That's want to motivate you. Why do we need to go back to atomistic representation sometimes? Because at this level of coarseness, you might not be able to see detailed properties and phenomena, including water permeation across the membrane. Coarse-grained waters are simply too big, and they are not charged. That's another problem associated with them. And we were not able to see any water permeation or pore formation, what people consider a pore formation in this kind of description. So what we needed to do was to go back to an atomistic description. So now this is the second half of your tutorial. So going from an atomistic description to a coarse-grained description is rather trivial, because you know that these 4 atoms are going to be grouped into one coarse-grained bead. So you reduce their representation. It's very simple. You find the center of mass of this guy, and that would be the position of your coarse-grained bead. When you want to come back, you have one coordinate, and you need to grow 4 different atoms into that coarse-grained bead. So it's not a trivial problem. There are multiple solutions for that. So you have to have some ways of coming up with reasonable atomistic structures for that kind of transformation. So there are lots of different kinds of tricks of doing that, and one of them is something that you will do today. So we talked about the coarse graining, so now we want to have a reverse coarse graining or fine graining, if you wish. And as I pointed out, there are multiple ways of placing 4 water molecules into this bead. So we have to have a way to facilitate that, to make sure that the chemical structure of the system will be reasonable for our atomistic simulation, and that's one of the protocols that we will use today. So just as again, for this particular system, we are going to, for those elements whose structures are well defined and we want to preserve those structures, we do reverse coarse graining. That's the case for the protein, how they aggregated. You want to preserve those configurations, and we reverse coarse graining protein and lipids. But for the case of ions and waters, usually you don't need to reverse coarse grain this. You can rehydrate and resolvate and add salt to your system again. So you don't usually reverse coarse grain these more fluid, less structured compartments or components of your simulation system. Okay, so this slide captures the way we did the reverse coarse graining. So or the protocol that was done in this study, we started with an atomistic description. We coarse-grained the system. Then we were able to do microsecond timescale simulations to see changes in the configuration, in the structure, in the conformation of peptides, how they aggregate. Once we have captured those large-scale, slow phenomena in the simulation, we were interested to know how these particular configurations characterized by coarse-grained simulations would behave in an atomistic system. So that's when we start to do reverse coarse graining. As schematically shown here, you will initially, when you map them back or project them back into atomistic descriptions, you might have really bad structures for this. So the initial mapping doesn't give you a sort of very nice structure. And at this stage, using different protocols, we try to refine this structure into a chemically sound and reasonable structure. And once you do that, after sort of what we are doing here is a kind of simulated annealing of this bad atomistic structure in order to relax it into a reasonable shape. What we are doing here is actually to constrain. So for example, this atom is going to be mapped to these 4 or 5 atoms up here. So we are going to constrain the position, the center of mass of these 4 or 5 atoms here to the position of the coarse-grained bead while we are doing the initial simulated annealing. So we have all these constraints acting on these groups of atoms, keeping them close to the center, to the position of the coarse-grained bead that represented them in the coarse-grained representation during the initial phase, so that they don't go anywhere, they stay where they're supposed to stay, but we allow the other degrees of freedom to relax in order to have a reasonable structure. That's one protocol of doing it, and that's what you will be doing today. So once you have this reasonable structure, then you remove these constraints, and then you continue your atomistic simulations, observations. Another thing that we do here, of course, when you are doing your mapping, you have to have a file that allows you to map back this into the same. You need to have a sort of initial conformation also for the groups that you are going to build into these models, right? So you know that you need to have a nitrogen, a carbon, and another carbon here connected to the next group. So the internal conformation or intramolecular arrangement of these groups also could be different. And in order to optimize this and to have a faster convergence into a reasonable model, what we do is that we save the conformation that we use initially for this particular group, coarse-grained. Before we coarse-grained, we save this particular conformation of these 4 or 5 atoms here, and we use the same conformation when we build it back into the coarse-grained model. Right? So that way we can also help faster convergence during this step. So that's another step that you might notice during your tutorial this afternoon. Okay, so that's actually what I already described on that slide. So in order to map back coarse-grained beads and then resolvating the system, we do 5,000 steps of minimization and then simulating annealing for about 20,000 steps, allowing the system to relax. During this step, of course, we are constraining atoms to the position of their representative coarse-grained beads. And that has been pretty successful. So, okay, so you go from a coarse-grained model. Again, you run a coarse-grained simulation and you take snapshots from your coarse-grained simulation where you think major things have happened, and then you examine them using a finer pair of glasses using atomistic simulation. So, and then you can start seeing that actually, now you can see there is a lot of hydration in the membrane region. So here you don't see any penetration of water. I mean, there is one exception here, but otherwise you don't see water going into this defective membrane, whereas when you come back to the atomistic simulation, you see a lot of defects. So these are some examples of water defects that are introduced into the membrane by these antimicrobial aggregates. So again, details here do not matter, but again, this is one example of the kind of phenomena that you might be interested in. How is this membrane defective as far as water permeation is concerned? But you cannot see at the level of coarse graining, and you need to move back and forth between coarse graining and atomistic simulation in order to capture that. Not a real multi-scale description, but by using different scales, you can really get the best of both. In one case, you describe very slow phenomenon, large scale kind of structural changes, and then you come back to a more fine-grained description to see what those aggregation or aggregates might, how those aggregation or how those structural changes might become mechanistically important. So these are water sort of defects that are formed using very irregular kind of conformation of the peptide, which is again completely against what was in the literature for a long time. I think immediately after this book, there was some experiments suggesting that there is a large degree of disorder for these peptides, which agrees with what we observed. And this is just to highlight conformational variability and disorder of the peptide in the membrane in this case. And here you can see a nice way of showing how much hydration you get into the membrane due to the presence of the peptide. Anyway, so after this work, we are suggesting that actually the main effect of this peptide is really introducing, in a non-specific manner, defects into the membrane, and that's how it sort of affects the cell. So non-specific general operation. Okay, so today you will use the lipoprotein example. This is what you use in your tutorial. So that's the main part of the tutorial. But in addition to this, you have a couple of additional examples in the tutorial, one of them dealing with coarse-graining a simple membrane, another one dealing with a transmembrane protein inside the membrane. So if those cases interest you more, you might want to play with those rather than the lipoprotein. So lipoprotein is a sort of little patch of membrane inside a belt of a protein. This is this lipid bilayer could include cholesterol and other kinds of lipids in your blood. That's how they are carried in your blood. And there is a large interest in knowing and characterizing the structure of this guy and the size of the particle here in this case and how they assemble. But of course, I mean, it's not an easy thing to crystallize experimentally. That's why modeling has been used a lot here to test and to come up with models, structural models for these particles. So you see we have the atomistic representation here and the coarse-grained representation. You go back and forth between these and you do some simulation on these today. Okay, so now I think we sort of covered most of the theory and how really this is done. But one thing that is important, I think, is to know what files are involved. This is something I wanted to talk about a little bit yesterday. Very briefly looking at the files that you're using for your simulation, because these are exactly where atomistic description, coarse-grained description, parameters are saved. You need to know where the information is saved in case you want to change, in case you want to use your own coarse-grained description or whatever. So that's why I would like to spend a few minutes just to look at the major files that you used already yesterday for your exercise. Okay, so let me start with the structure. So that's just to give you a brief in case you haven't looked at this yourself. So this is the coordinate file that you usually use for your simulations. This is so-called the PDB file. We have for each atom, we have, in a PDB file, you have the coordinate of the atom, XYZ coordinates. We have the residue they are associated with it, and you have the atom names. Okay, so in case. So these guys don't have any chemical meaning. We only know that there is a methionine residue in this protein that has atom names of this sort, and these are the positions of those particular atoms. So you need to have a topology file in which you connect or you derive the sort of chemical properties of these individual atoms for that particular residue in order to be able to use the right parameters. So yesterday you remembered that we need to specify all the bonds, all the bond angles, all the dihedral angles for each kind of interaction. You need to provide the right parameters. And that's something that is not present in the PDB file or in your coordinate file. So you can see for all the residues in your protein and later for your water. This is a ubiquitin in water that you use for your example. These are water residues individually specified as T3P, for example. So this is your PDB file. That's a sort of simple case, easy case. Then what is important also for your coarse-grained kind of tutorial today is the topology file. That's where you specify the topology of the system. That's it. So here, I mean, this is an example of the charm topology file. Here it doesn't matter what you are using. So you have the masses of all the atom types. Now we are introducing the concept of atom types. This is actually pretty important distinction between atom names and atom types. Atom types are chemical types for which we need to have the correct mass, we need to have the correct parameters as far as the interactions are concerned. These are the interaction sites. So every atom name in your PDB file should be mapped onto one of these atom types in order for you to be able to derive the right parameters. So the masses are here, and then later for each residue, you can see, for example, for the case of alanine, we can say there is a residue called alanine. This guy has these atom names, and every atom name is connected to an atom type and its partial atomic charge. So if you want to change something in the file, this would be, for example, if you want to neutralize all alanines in order to do a test simulation, the topology file would be the place to do this. So you have atom types, and you have the charges here, and then later for this particular residue, you have all the bonds. Essentially, the connectivity within this residue is defined here. CP is connected to CA, N is connected to HN, etc., etc. So your connectivity is defined here. Now all the bonds are defined here. Angles and dihedrals are generated automatically, usually in this particular file here, auto angles, dihedrals. And by default, the first and last residues will be capped because it's assumed to be a protein. This whole thing was evolved around protein simulation. That's why everything is called a residue, including water is a residue, lipid is a residue, any ligand you have is a residue. So the connectivity is defined here, so you have all of the information about the connectivity here. So if you're using a standard residues, you don't have to do anything. If you want to add your own ligand, then you have to define this ligand here. So now, instead of using atomistic description, you can have a topology for alanine that has only two atoms. One is a backbone atom, one would be a sidechain atom, or B, I should say. And then in your PDB file, instead of having 10 or 15 atomic coordinates, you would have two sets of coordinates for these two bits. That's the only difference. That's the only place that you sort of essentially change the representation that you use for the system. Of course, the force constant parameters are going to be different as well. But as far as the topology and the representation that you use, you will be changing your atomic representation because you are now looking at the center of mass of groups of atoms, and you will be changing the topology of that. You will have different number of atoms and different connectivity. So this is for different side chains. You have all this information stored here. And that goes on and on for different kinds of residues in the protein, for water, for ligands, heme, and other kinds of molecules that you might be interested. And of course, as I pointed out, if you have your own molecule that is not in here, you have to build this topology information in here so that you can use it to generate the protein structure file. So for those of you who might be actually interested in doing protein simulation, there is a, let me go back. This one thing I think is very important because it also connects to the fact that you don't have polarization and you're using fixed charges. So histidine is a particular side chain that can have different protonation states. We have quite a few of these side chains actually. And as we discussed yesterday, we have a fixed representation for the charges in our classical description. This is not the problem only for proteins. In general, when you have a molecular system, you might have polarization in general and protonation, deprotonation of certain sites. And that's something that is unfortunately not implemented in our classical description. So if you have a change in the protonation state and a charge state of part of the molecular system, we will not have it included in here. But in some cases, so you have to make up your mind, what is the most representative form of that particular residue of that particular chemical group that you want to use. You want to use a positively charged amino group there, or you want to use a neutral form. So this is usually done based on pKa calculations for these side chains. In most cases, you have clear pKa's. For acidic residues, you have pKa's of 3, 4, meaning that at a neutral pH where you usually simulate your system, these are going to be charged, deprotonated. And for basic side chains, we have very high pKa's, 10, 12, meaning that they are going to be charged too at a neutral pH. But for certain groups, this becomes really tricky. For histidine, for example, the pKa of this side chain, it has 2 hydrogens, two nitrogens in its side chain, is pretty close to 7. It's actually 6.5. So it becomes really difficult to decide what is the representative protonation state. And even if you know the neutral form has a slight preference or sort of a slightly higher population, you don't know where to place the hydrogen. You can place the hydrogen here, or can place the hydrogen here. So it has 2 tautomers, so to say, 2 tautomeric states. So for that, you have different topologies, different configurations. Again, based on the local environment of that histidine, you can decide what tautomeric state or what protonation state might be the most representative for. So there are quite a few of these that you have to keep in mind in case you have them in your molecular system. But that's a good example of the problem associated with fixed charge or fixed protonation state simulation. So we have 3 kinds of histidines. HSD has a proton at the delta nitrogen. Then we have HSE down there here, which has a proton on the epsilon nitrogen. And then later we have the HSP, which is a protonated histidine. It has 2 hydrogen, 2 protons, and it carries a net positive charge. So there are many residues of this kind that you can decide which one is the best one to choose. Okay, so then you have the topologies here. You see there is no information about the coordinate, but all the connectivity information and all the charges are defined here. So you get your PDB file. Actually, one more point about water, which is important in case you are considering different force fields. So water is also there. So water is also defined as a residue with 3 atoms, and this is the so-called P water. So it's sort of 3-point model for water. There are more than 100 different models for water, depending on what you want to describe. If you are after diffusion constant and diffusion properties of water, this is not the best model. If you want to have the dielectric effect of water, you have to use a different water model. But in our limited description, we need to go with one form of water. So in CHARM, they are using TIP3P water, and everything else is fitted to interaction of different groups with water. So you cannot use a better water model in CHARM if you are running your simulations on a protein with CHARM. For example, there is another water model. I think yesterday someone was asking SPC, which has better properties as far as diffusion is concerned. But you cannot mix it with CHARM, because in CHARM, all the energetics are fitted to interaction with this particular water. So if you introduce a different water model, not all, maybe your diffusion properties are better for bulk water, but all the interactions with the protein are going to be messed up. So there are this is important for the particular case of CHARM case, but I can imagine there are other similar considerations that you are trying to choose the right force field or trying to mix and match different kinds of force field. You have to always keep that in mind. In some cases, even the conditions that you use for your simulations should be the same. For example, all of the testing in CHARM has been done using a certain cutoff, 12 angstrom, 10, 12, and a certain kind of time step, 2 femtosecond time step. So in order to stay consistent with the conditions in which the force field has been optimized, you should try to use similar conditions. Don't just change it and say, oh, I'm doing better. My cutoff is now much better than 12. I'm using 20. So my simulation should be performing better. No, not necessarily. You might actually even use a different level of theory when you are deriving parameters. They have used Hartree fog and different levels of MP2. Maybe you can say, I can do much better coupled cluster kind of calculation or much larger basis set. Yeah, so in principle you're right, but then you become inconsistent with the rest of the force field. So we have to try to stay consistent with the rest of the force field when we are developing new parameters here. But again, sometimes, again, this is what has been done, has been tested. We try to stay consistent, but in some cases you know that because the electronic structure is so affected or so wrong at this level of theory, I cannot calculate it using the standard protocol. I have to go into higher levels. So if you have really strong justification for doing that, good. But otherwise, we should try to stay as consistent as possible. Okay, so we have the topology in here. We have the coordinates where we have the residues. And then what you did, one of the steps that you did yesterday, hopefully, was to generate the protein structure phi. That's the PSF was generated using the PSF gen for NAMD. So what you do here is to look at your PDB file and say, I have this residue called methionine. Let me look at the topology file, see what information I have for methionine, and you dump that information into this PSF file. Then you go to the next residue, get the topology for that residue, dump it into the PSF file. So the PSF file will contain all the topological information, connectivity, the number of bonds, atomic, atom names, atom types, and charges for the whole molecular system. So in the beginning, you have 7,051 atoms. They're all described, what segment they belong to, what residue they belong to, what is their atom name, and what is their atom type. And you will see that we will use this atom type information to derive the correct parameters for interaction between these atoms and other atoms in the environment. So these have chemical meaning, whereas these are arbitrary atom names. That's going to be the partial charges for all the atoms, and these are the masses. So now, if you want to artificially, and I sort of ask you to do so in your simulation, try to put it under extreme artificial conditions to test hypotheses. For example, if you want to change the mass of some of the atoms in your system, the PSF file would be the place to work with. Maybe you can make this atom 100 times heavier so it doesn't move, or see what happens. Or you can actually, later, initially you have the information about individual atoms. You can see this is the first block. Later, you have the information about the bond. You see, for each atom, we define an index here. And then later, in the next block, we use these indices to define the number of bonds and number of angles that are present in the system. So we can break one of those bonds very easily by just coming to the PSF file. Too many other ones. So this is the bonds block where you define the bonds. Say, I mean, there are 5,117 bonds here. Atom 1 is connected to 5, 2 is connected to 1, 3 is connected. So you can delete a bond easily by just editing the PSF file. Or you can add an artificial bond here for yourself, really testing something. If you introduce new bonds, of course, you need to have the correct parameters for that particular bond, right? So that's the only thing NAMD needs. You generate this PSF file, you have the coordinate in your PDB, and all the information about the bonds, later angles, dihedral angles are stored here. So the only thing NAMD needs, NAMD doesn't need the topology file. It needs this one, this PSF file. So it comes here and says, okay, you are telling me atom 1 is connected to 5, goes up there, finds what is the atom type for atom 1, what is the atom type for atom 5. So I have these two atom types interacting with each other through a bond. I need a spring force constant, and I need an equilibrium value for this interaction. If you give me that in the parameter file, I continue. If you don't give me that, I stop. That's how NAMD works. So now after defining all the bonded information here, and then the last file we need is the parameter file. There, all the parameters for the potential energy terms are stored. So this is, they usually come in pairs. The parameter file, this one is for all atom description, version 27, protein lipids, and it usually comes with a topology file that has a very similar name. So we usually use this in pairs. So here, as you can see, again, for every pairs of atom types, see, that's the bond part. So we have this is reminds you of the shape of the potential. You have the harmonic potential here. K is the force constant, B0 is the equilibrium value, the units. And then for each pair, you have the force constant and equilibrium value. And this goes on and on. As you can see, the force constants are on the order of hundreds of kilocalorie per mole. This is a very stiff degree of freedom in a molecular system. Later, you will have the section for bond angles in a very similar fashion. That's your potential for functional form. And then for each three groups of three atoms that are connected to a bond angle, you have the force constant. I don't want to confuse you with the Urey-Bradley sort of additional term here. Let's not worry about that at this time. So the force constants are on the orders of tens of kilocalorie per mole per radian square. In this case, softer degrees of freedom. And as you can imagine, you have a similar set of parameters for dihedral angles. So dihedral angles are described using a cosine function here. And here for each dihedral angle, you need to have three parameters: force constant, periodicity, and the phase. And this is exactly what you have here for each set of four atom types. Okay, so that's it. That's it. I mean, the only difference between dihedral angles and bond angles and bonds is that because you have a cosine function here, you can have a series of these cosine terms really describing complicated, complex forms of dihedral potential for you. So you can have multiple definitions and multiple lines for the same set of atom types in this case. And you can have wild types. Sometimes you have a star there that means it's not a star, it's an X actually. So that means a wild type. So if you don't have any explicit definition for the dihedral angle, for that particular dihedral angle, the program might actually use this wild types to get this something. So here, for example, these are wild types, wild cards, I'm sorry. So that's it. And then at the end, you have the information about non-bonded interactions, Lennard-Jones. The charges are already stored in the PSF file. You have them. And the Coulomb potential is pretty straightforward. So for the, this is the improper angle, which you can use in forced planarity, or certain types of shapes, intercenters. But then for the Lennard-Jones, you need to have epsilon ij and r min ij for each pair of atoms in your molecular system. So the way it's done, it's sort of, it derives these pair potentials or parameters based on the combination rule. You specify epsilon and r min half for each atom type. And then by looking at the average of the two R-mean half values, you get the R-mean ij. So that's the parameter for that particular pair potential. You just add this and get the average of these two. And for the epsilon, for the depth of the energy well, for your van der Waals potential, you look at the square root of the product of the individual epsilon. So you use the combination root to derive this. So this is just to give you an idea of where the information is stored in case you want to change it. And again, as I pointed out, the only difference that between the sort of coarse-grained representation that we use today and atomistic representation is at the level of the topology, parameters, and the coordinate file that we use. The engine, the kind of functional forms that we have there, everything is actually the same. Okay, so any questions actually for this particular files and how we use them? Okay, so at the end actually I just talk about it. Another sort of approach that we started actually in the group to deal with the problem of time scale, and that's a sort of, we're taking a different angle at the problem. Okay. And I guess, I mean, this is a, so again, remember, the whole problem is the time scale. Or the major problem here is the time scale that you want to be able to describe. So we talked about using bias simulation to accelerate certain processes, to excite certain degrees of freedom in the simulation system. We talked about coarse-graining and how we can reduce the number of degrees of freedom, how we can use software potentials to make things happen faster. So another kind of approach, and I think just take 5-10 minutes because I think it might be interesting and might inspire you for similar for other molecular systems, is based on this sort of concept of fragmentation, what we call. So one example in which we have applied the system is the case of membrane bilayers. We talked about this. We have the head group, we have the core of the bilayer, and distinction between polar interaction of the head groups with water and hydrophobic pairs that don't like water results in certain type of structures that you have with bilayers. So oftentimes there are lots of proteins that need to have atomistic description of the lipid molecules because they specifically interact with certain type of head groups. And sometimes actually these head groups are necessary for their proper function. Just to motivate you, in your blood you have a lot of coagulation factors right now, but they don't form a clot because you don't have the right lipid molecules on the surface of the membrane. They just keep floating in the blood. As soon as you want to form a clot, or your body wants to form a clot, there are certain types of lipids that come out from the inner leaf plate to the outer leaf plate, and their exposure to the surface is the signal for these proteins to bind to the surface, and once they bind to the surface, the whole coagulation cascade starts. So the whole coagulation cascade depends on the specific interaction of this head group with this particular protein. And that's what we wanted to do for this particular case to describe. But the problem is that if you want to look at this process in using atomistic simulation, binding of this protein to the membrane, it doesn't happen. Time scale problem, right? The same thing that we discussed. So we have another problems of similar kind. So the same thing happens if your protein is undergoing very large conformational changes. Again, this might not happen because one problem here is that these lipid molecules are very slow, right? We looked at diffusion and answer on these guys. This is on the order of 10 to the minus 8 centimeter per centimeter square per second. So now, one thing that we did a while ago was to use SMD. So we keep the bilayer and we apply a force to the center of mass of this whole object, and we slowly pull it down. We ran fairly long simulation back then 4 years ago, combining bias simulation and further equilibrating the system to see the process taking place. Sometimes it was successful, sometimes it was not. But we managed to have one single nice simulation in which we described the insertion of this protein into the membrane, again using biased kind of simulation. Very nice model, we published it, but we never managed to collect enough statistics on this process and to find a specific binding site for that particular lipid. So it was a problem. We needed to stay atomistic because otherwise we couldn't describe the specific interactions, but the whole process is too slow. So we have a lot of proteins of this kind. They are known as peripheral membrane proteins. They only bind loosely to one surface. So then we came up with this idea that, okay, instead of coarse-graining the whole lipid, in which case you lose the atomistic details, why not going in and sort of liquidify the core of the bilayer? The core of the bilayer, as far as interaction is concerned, is just a bunch of hydrophobic molecules, right? Instead of having these long lipid tails that entangle and they make the lipid molecules very slow, we can actually replace the core of the bilayer with an organic solid. Hydrocarbons, still liquid, still hydrophobic, but it gives you much more fluidity in the core of the bilayer. Still everything is atomistic. You don't have to worry about coarse-grained approximations, etc., etc. But by doing this, we are able to increase the mobility of the head groups that are still atomistic, right? So the important region of the system is still described atomistically, but in the core, those guys are represented by fragments. It's like you go in and chop those tails into little pieces. They become much more fluid and much more liquid. And by doing this, without worrying about coarse-graining, all the problems, we have been able actually to increase the mobility of the lipid bilayer significantly by 1 or 2 orders of magnitude. So this is what we did here. So again, we had to optimize. See, most part of the tail is chopped into little pieces, but we kept a few carbons of the tail. We had to calibrate this. That doesn't matter. But actually, by doing this, we have been able to see the formation of bilayer, something that people only could do in coarse-grained description. Now we are looking at formation of the bilayers, and May was actually one of the people who worked on this. This is his simulation. So we have been able to see the formation of the bilayer starting from a completely scattered configuration of these short-tailed lipids in 22 nanoseconds. And I think that's really the key here. So by doing this, we can now repeat this simulation multiple times and collect a lot of statistics. We can do this simulation forming the bilayer around your protein or around your object. Look at the preference of different kinds of head groups for your protein, find binding sites for the protein. So this has been extremely actually successful in terms of capturing this or increasing lipid diffusion in membrane-like structures. This is the same thing that I showed you in that movie. You can see how nicely and how fast lipids converge to the two leaflets in 10-20 nanoseconds using atomistic representations. If you look at the distribution of different chemical groups in the head group region, it matches very closely what you get in a full membrane representation. The face of the lipid bilayer is very similar in full representation and what we call highly mobile membrane mimetic HMMM. Even the position of the counter ion is matching very closely. We only have actually wider distribution here because they fluctuate more, they are more flexible, and this we have been able to fix by adding additional constraint to the system. So if you look at the diffusion, this is a full membrane representation in 10 nanoseconds. You can see lipids just locally fluctuate, whereas this one is our new membrane model in 1 nanosecond, 10 times smaller, but you see a lot of motion here. Of course, we can quantify this. So these are the diffusion constants for different models. These are the full conventional membrane models that people have been using. And you can
going to this HMM representation and other tricks, you can increase the diffusion constant by 1 or 2 orders of magnitude. This is a logarithmic scale. So you increase by 2 orders of magnitude the motion of the lipids without losing the atomistic detail of the layer. That's really the key point here. And now in the simulation we had to do using biased Coulomb simulations, and sometimes working, sometimes not. Now we have been able to do this 10 times, really, in our cases, seeing binding of the protein in equilibrium simulations. Now we are not applying it. This is very nicely to the membrane, converging to a very similar configuration. So we are very proud of having N in our paper, like experimental. Okay, so, and again, I think this is another case just to show that this system is working for different kinds of peripheral membrane proteins that bind only to one surface. This is actually a nice way of, a nice model if you are interested in binding of or absorption of proteins or other objects, drugs, for example, or any membrane-associated phenomenon that is slowed down by slow lipid diffusion. So we can really nicely see what happens at the surface of the membrane. We can again actually see how membrane binding changes the conformation of the protein. These red residues initially form the cluster, but once they see the membrane, they open and insert into the membrane. This particular protein is actually something that is involved in integrin activation. Again, the details do not matter, but we are able to even characterize membrane-associated phenomena. Okay, so let me skip this because these are unnecessary details. The Helix insertion problem that we did using coarse-grained models, and I told you using atomistic simulation it would be extremely expensive. We can describe here, so you can see, but again, please pay attention to this 12 nanosecond. This is extremely fast for what we think is a fairly realistic representation of the membrane. So those copies of the helices are inserted into the membrane. It's a very nice way of looking at the effect of thickness of the membrane on the tilt angle of the helices and how they interact with each other, what is the optimal thickness of the membrane to have optimal interaction between these two helices. Because everything is happening 10, 100 times faster. And still you are preserving atomistic details of the head groups that are important for, that might be important for your particular protein. So, okay, so that's, and again, as far as the properties, something that you need to remember is that the mechanical properties of this membrane representation is of course different. So in a full membrane, you have different compressibility. The compressibility coefficient is pretty high, whereas in this representation you have a much lower by design, because it's supposed to be looser, more flexible, and moving much faster. So that's one thing that we don't agree with the full membrane representation. But if you look at the electrostatic properties of the system, so membrane dipolar potential, for example, we are able to capture the correct behavior, which is important for reflecting actually what the degree of hydration of in the head group and the electrostatic interaction with the protein. That is right on. And of course, when you look at free energy of insertion of different amino acids, again, becomes important for protein interaction with this membrane. We seem to be doing very well. So in the core of the membrane, we have some differences between our model and a full membrane representation, because the core is done using an organic solvent, too much fluidity, we have much higher entropy, and usually they are sort of over-stabilizing hydrophobic residues in the core of the membrane. But in the head group region and the initial part, they are right on using the simplified. Anyway, so that's actually that. I thought actually this is a sort of fairly different kind of approach toward the problem of time scale, describing the system using fragments, still everything mechanistic, everything consistent. You don't have to worry about those problems that arise, and you have a multi-scale kind of approach and description with different kinds of lipids. And actually, one additional thing that we have been implementing in NAMD and DMD is that once you have your phenomenon described very well using this different representation of the membrane, you can slowly grow the tails and make it a full membrane in a stepwise manner. So we grow them 2 carbons at a time, simulate them a little bit, relax them a little bit, using a sort of combination of multi-carbon type of selection of the solvent in the middle. We essentially are eating the solvent in the middle to grow the liquids and slowly make a full membrane representation of the system. So that's it.
Okay, good.
Advertise a little bit for the kind of phenomena with this, just to give you an idea for what systems we are using them, the variety of transmembrane and peripheral membrane proteins, drugs, viral peptides that bind to the surface of the membrane. And so if you have anything, interaction of some materials with the membrane, you might want to actually consider this. This is really useful. Okay, so I guess we are done.